Good evening. Is working? Good evening. Thank you. Thank you very much for coming, and thanks a lot for the invitation to the Congress. I'm really enjoying the speakers. I did it today in the morning, and I hope this couple of hours, an hour and a half, I think, Dr. Oser is following me, will be also enjoyable for you. The title, I think, in general, titles said it all. Does the glutamatergic system play a pathophysiological role in mood disorders? This is an hypothesis, and I would like to show you why this hypothesis was generated, and then to show you as well a set of clinical studies that we conducted in order to try to, in order to confirm or deny this point of view. This is Beethoven, I think. And I'm a full-time employee of Johnson & Johnson Pharmaceutical Research and Development now, although the work that I will share with you today was conducted at the National Institute of Mental Health in Bethesda, Maryland, in the States. And it has no relationship with the work that I'm doing now for the pharmaceutical company. And also I would like to share with you that I will discuss off-label indications according to the FDA for some of the medications that we tried in patients with major depression, was treatment-resistant major depression, and those are Riluzole, memantine, and ketamine. And I also want to disclose that I received funding from NARSAT in the States while at the NIH. So this is my first and last try to speak Turkish. The title is Duygu durum bozukluklarinin fizyopatolojisinde glutamatergic sistem rol oynuyor mu? Bu varsayımım klinik verilerle irdelenmesi. I'm guessing that I did it. Okay. So I would really like to learn more actually, but here we go. I would like to share with you then this slide that I have prepared to understand what is the problem. So the problem is that from 100 patients that receive treatment for major depression, only a portion of them are responding. And I'm showing you the amount of patients that are remaining in different treatments. So you can define these responses to antidepressive treatments in different ways. And I have defined here first, second, third, and fourth line. In the first line, as you're aware, monotherapy is in general the most common initiation for drugs, in general SSRIs. A second line of treatment consisting on a second medication, or sometimes to switch if the patient didn't show any response with the first medication. In general, the strategy is to change to a different SSRI or to start an SNRI. And then by that time, only 2/3 or 60% of the patients has responded to SSRIs, and you have still 40 to 50% of the patients that will still require this second line of intervention. From those, you have, again, half of them that will not respond to the second line, so you have 22 to 35% of the original 100% that will require an augmentation, if not a different add-on on a different switch. As you know, the add-on in general is with mood stabilizers like lithium or valproate, sometimes thyroid supplement, or you can change the augmentation, or you can change to a different kind of antidepressant again. There are a lot of options. And again, half of them, and here the statistics are very rare, so it's hard to find this data. But what I found is that between 1% and 28% of the patients will still remain non-responding to these treatments, and there is when you have to start the artistic treatment of patients with new combinations, and at the end we know the effectiveness of ECT, and now repetitive transcranial magnetic stimulation has been approved as a treatment for this population. We have other options as vagal nerve stimulation, and then, as was discussed today in the morning briefly, in an experimental fashion, still deep brain stimulation. And so this is where we are with in terms of how we can treat patients that are resistant to major depression. So why these patients are not responding? Mood disorders likely arise from a lot of different susceptibility and protective genes and that are interacting with environmental factors. So therefore it's very difficult to have an homogeneous group of patients to be treated. And then we know that the phenotypic expression not only involves the disease, the mood, but also involves cognitive, motor, autonomic, endocrine, and sleep-wake and abnormalities. And therefore, again, a lot of different subpopulations, probably more like a syndrome instead of like a particular disease. And finally, because of the same, there is a profound need to understand the pathophysiology of this disorder. And here is where we think that the glutamatergic interventions may have something to say. So the objectives of today's presentation are to review how severe mood disorders might be associated with brain morphometric changes, to summarize the therapeutic effects of mood stabilizer that can be associated with increase in structural plasticity. Today, Dr. Meltz in the morning mentioned he has some data regarding clozapine that is now being generated, and changes in neuroplasticity and probably morphometric changes in patients with schizophrenia. And to present clinical evidence, preclinical evidence demonstrating that glutamatergic system may play a role in this pathophysiology, and to propose how glutamatergic neurotransmission could play a role in the pathophysiology of the disorder in humans, and to review a series of clinical studies that we conducted targeting the glutamatergic neurotransmission. So a couple of years ago, we published a group of papers trying to understand how we can better comprehend the pathophysiology of mood disorders. And basically, there are two approaches, from the top to the bottom and from the bottom up, trying to understand how medications that are useful in the treatment of these disorders, as lithium and valproate, for example, in the treatment of bipolar depression, act, what is the mechanism of action, and from there identify new targets. And second, based on the knowledge obtained from what we think is the pathophysiology of depression. So the approach that I'm going to share with you now is a combination of both, actually. And this slide is very busy, but I think it's informative in the sense that it's showing all the information that already exists in the literature regarding changes in morphometric structure in patients with severe mood disorders. So I have summarized here the main changes, and these are the well-known and described also in schizophrenia, like increasing the lateral ventricles and third ventricle and the volume ventricle ratio, decrease in basal ganglia volumes, decrease in frontal lobe volume and area. We will stop here for a second in the next slide. Temporal lobe volume and area, and then some findings indicating increasing activity and size of amygdala and hippocampus is still not very clear the direction of those changes. So how these changes were generated? So several investigators have looked into the brain of patients after they have been when they obtain these brains, and actually they have found, very interestingly, that the volume and the cortical thickness has been decreased in these patients as a correlate with the morphometric changes observed with neuroimaging, but also that the number of neurons, and here it's very important that these are layer specific, pyramidal neuronal density and layers 3 and 5, for example, in prefrontal cortex in patients with bipolar disorder. The same has happened with the number of glias, and with the number of glias is the density, again, in the very same areas, prefrontal cortex and some basal ganglia. So we don't know why these changes are there, but now we know that they are. So that's the first step. This is a description of the data, and now we can try to hypothesize why this is happening. And one of the reasons is because observing the decrease in glial cell number and density, you can speculate that glia having an important role in regulating synaptic plasticity and actual glutamatergic concentrations may be involved in the energy homeostasis in the central nervous system. And in fact, they can produce some trophic factors that may put these synaptics may have the synaptics functioning well in the brain. So this is the glutamatergic system. It's a complex slide, but I will try to point out to the essential. And actually, I selected this slide because it's complex. The glutamatergic system is a complex system. We have genotropic receptors. The genotropic receptors are ion-gated receptors. Basically, they are channels in the membrane of the cell that open and close when they are stimulated. And the stimulation of these channels is with glutamate in this particular case. But you can dissect with some chemical entities which channels are activated with which chemical entities. And they have been identified at least 3. And those are the NMDA, N-methyl D-aspartate channels, the AMPA channels, and the kinase channels that are not shown here. So there are 3 kinds of genotropic channels. But to add to the complexity, each channel is composed by 4 subunits. And these subunits change according to your developmental age. For example, GUA subunits are, if I remember correctly, some of the subunits are not present at the embryo stage, but they are early in the development. So you have that level of complexity. But on top of that, there is a different set of receptors that also interact with glutamate, and these are the called glutamatergic metabotropic receptors. So these receptors are transmembrane receptors, but this time they are not a channel, but they are a protein that push another protein in general for these receptors is always a G-coupled protein to the receptor that activates this trimeric unit, and that produces the action. And that action could be inhibitory or stimulatory downstream. So this is where we are. And this is a simplification of the prior slide, but will serve the purpose of this talk. So here we have again the glia and the presynaptic neuron, the postsynaptic neuron, the ion channels. This is the best I could do with PowerPoint, better than handmade. And the glutamatergic metabotropic receptors, and they are distributed unequally in these pre- and postsynaptic neurons, and some of them they have stimulatory functions, and some of them they have inhibitory functions. So a lot of data has been generated to support the role of this glutamatergic neurotransmission activity in major depression. Very early on, the use of amantadine, an antiviral that also was used for Parkinson's disease, showed that in animal, a well-known model for depression, the force-swim test in rats, was positive with this medication. And later on, AMPA kinds, which are medications that target the AMPA receptor in the glutamatergic synapse, also have activity in these very same models. So, as I was mentioning before, we have the demolition of the glutamatergic system may have an antidepressant effect, and this is through the NMDA receptor antagonist, and has been investigated in all these models of depression. I will not stop there. Very importantly, a single dose of the NMDA antagonist ketamine, I mentioned at the beginning, in some rats interferes with the induction of behavioral despair for up to 10 days after the regional administration. Very interesting data. I will come back later to that. And that repeated administration of antidepressants brings also alterations in the expression on NMDA subunits RNA messenger. So this is just to show you some data, clinical data. This is ischemia induced by mediocerebral artery occlusion. And you can see that when these rats were pre-treated with lithium, so a new element in the conversation, lithium, the size of the infarct is much reduced after this chronic treatment for 2 weeks. So how is that could be related with depression and with glutamatergic activity? A similar effect is observed with infusions of quinolinic acid, which is an NMDA receptor agonist. So therefore, there may be a connection there. But in addition, we know that this molecule, lithium, that is a simple cation, can protect the insults of glutamate and NMDA receptor activation, also increases in calcium, and also protects the rodent in vivo models as the middle cerebral artery occlusion. So we are shifting a little bit, I'm aware of that, and we will come back though. So mood stabilizers may differentially regulate AMPA receptor. trafficking. So understanding how the mood stabilizers may work may give us a hint of a target, an identifiable target to try with new medications. So as a confirmation of that fact, it has been shown that lithium and valproate reduce some of the subunits that I showed you that are component of the NMDA receptor after they are treated. And very interestingly, and I know that the slide is a little bit busy, when you separate this mood stabilizer into those who has an antidepressant and anti-mania effect, and in the antidepressant side I will put here lamotrigine and riluzole, and then the antimanic ones like valproate acid, you have that the direction of the change is different. With the antidepressants, you have an increase in the expression of GLUR1, these subunits of the NMDA receptor, and the same happened here in this second model, on which valproate subunit of GLUR2 this time is decreased, while the other ones, GLUR1 and GLUR2, are increased with these other medications. And here a very interesting piece of data. I understand that there's a lot of preclinical data, but it's very interesting. These rats were tested in the first swim test model. This is a test on which you have these animals swimming, and the immobility time is measured. The immobility, when the rat swims more, it's a sign that the rat is still fighting, and that has been associated with the antidepressant effect of SSRIs. It's very strange, but that's the way it is. And so a decrease on the immobility time, basically, which is an increase in the time that the rat is fighting when put in this tube to swim by itself, has a correlation with the antidepressant activity. And as simple as it sounds, it's one of the best models that we do have to predict antidepressant activity in humans. And that model has not been changed in a bunch of years. So this is where we are, and this is where we need to try to move forward. But considering the limitations of this test, it's still very interesting data. You can see here that imipramine reduced the immobility time, meaning that increased the antidepressant effect. But when you have an agent that blockades the AMPA receptor, like NBQX, you have an increase a little bit, almost to normality, of the time of immobility. And again, when you give imipramine associated with that agent, you have that the decrease is a little bit different. So part of the antidepressant activity of imipramine is mediated by AMPA receptor activity. That's the message of this slide. Similar there, I will not stop now though. This is the same, this time it's with chronic administration of actually, let me go back to this one. Very, very important piece of information that I missed. This slide is showing us the same effect with ketamine. Ketamine is this NMDA antagonist that has been used as an anesthetic in pediatric population in humans. And we can see here that ketamine also decreases the time of immobility, so it has an antidepressant effect. But that effect is blocked when you add ketamine and this AMPA receptor modulator. So activity in the glutamatergic system may be related with antidepressant effect that we observe in these animal models. The same data here is duplicated with chronic administration of ketamine. So let me change gears a little bit to the clinical correlates of this glutamatergic hypothesis. So as it was shown today in the morning, it has been observed early on in 2000 that lithium, that we are hypothesizing that part of its mechanism of action is mediated through the glutamatergic modulation, also increase the cortex in terms of volume and area in humans that receive the treatments for at least 4 weeks. And this is very shocking data. That is not that lithium is increasing this cortical volume, and this is not due to the swallowing of the cortex. The water was controlling this experiment actually. And then we also can see that these effects were not observed in white matter, so they are specific of the cortex. So there is something that is happening between the neurons and the glial cells in the cortex that is explaining the story. Is this associated with the findings that we found before about decrease of neuronal and glial cell and number in patients with major depression? Is this impairing that activity in depression or not? That's a very interesting hypothesis, and that's the one that we're trying to test. But even more interestingly, when you try to understand which of the areas are the ones that are involved in this increase in total cortex volume, you can find again that they're the very same areas. Prefrontal cortex, left trigeminal prefrontal cortex, also mentioned today in the morning, left hippocampus and thalamus. So if that is happening with lithium in patients that were treated for 4 weeks, more in a naturalistic fashion, what is happening. So this is a post-hoc analysis of a series of patients published by Wayne Drevitz before, but very, very interestingly, let me walk you through this slide. You can see that the controls has this level of volume prefrontal cortex. Here at 230, the number is not relevant, but to have a reference point. And then when the patients have major depression, they have a decrease on that volume when you compare the patients with the controls, as we have observed before. But very interestingly, bipolar patients that were receiving chronic lithium or valproate at the time when the images were taken, they have a decrease that is not as accentuated as the decrease in major depression. So something lithium and valproate are doing that are protecting the cortex of these patients of being reduced in the area. And when you can compare with bipolar patients that are here as the control group, that were off lithium or valproate, you don't see that benefit, and you see that they behave as the patients with major depression. More confirmatory evidence also is coming from studies from magnetic resonance spectroscopy that have shown that patients with bipolar disorder have an increase on N-acetyl aspartate. N-acetyl aspartate is a molecule that you can measure in the spectrum that you observe with an MRI. When you change the MRI view from, with a program of course, from the neurological image that we are used to into a spectrum, you can observe, depending on the potency of that magnet, of that MRI, you can observe different molecules. In general, you can observe glutamate, glutamine, GABA. You can observe choline or creatine, but also N-acetyl aspartate. And the important here is that this is the one that was increasing these patients. Why it's important? Because N-acetyl aspartate, or NAA, has been believed that it's a marker of neuronal viability. And therefore, when you have more in adult neurons, when you have more N-acetyl aspartate, those neurons, this is a simplification, those neurons are more resistant to have any atrophy. They are more ready to resist any insult. So here, confirmatory evidence that these patients, these areas, also have an increase of this neuronal marker of viability. Basically, that maybe that the volume of the neurons is higher. We're not saying that the number is higher, but the volume of those neurons may be higher. This is an experiment that we also conducted at the NIH, basically confirmatory, finding that N-acetyl aspartate and N-acetyl aspartate glutamate was lower in patients with bipolar disorder. These are patients that are not being treated. They are off their medications, and they are going through a period of depression in this particular case, which is very interesting if you think that precisely these are the levels that were increased when the patients were treated with lithium. So moving between these two ways of understanding the pathophysiology, from what the medications do, lithium valproate, and what do we think is the pathophysiology. So I'm going to come back to the glutamatergic story. Clinical evidence of that the glutamatergic system may be involved in major depression is abundant. And here I have listed the ones that was available until 4 years ago. But basically there is an increase of the GLX, which is a measurement of glutamine and glutamate and GABA sometimes. Glutamine ratio in patients with bipolar disorder. I have selected another one. Glutamine levels elevated in the CSF of patients that are depressed versus the controls, etc., etc. So the evidence is there, and let me show you finally what is the hypothesis. So all this preclinical evidence data that I have shared with you is to get to this point. So this is what we think may be happening. And of course, this is a simplification, but as a simplification, helped me actually, to understand what we were going to do with the clinical studies I will present next. So basically, let me show you a neuron working normally, normal affective functioning. So this is a neuron, this is a glutamatergic neuron that is sending glutamate here, and you have the genotropic receptors, the metabotropic receptors, and they are in a good tone. And then you have a very healthy cortex, and actually here with a good dendritic arborization, they are very healthy. And on top of that, you see a lot of synaptic buttons, here a drawing and here an electron microscopy photography. So then what is happening? We think that in major depression, this system is hyper-functioning. And therefore, I didn't tell you this part, but I will tell you now, when you have an hyperfunction of this system, you have an opening of these channels, and a lot of calcium is getting into the cell, will maybe, and I will show you in the next slide, that may be related with neural toxicity and atrophy. So in this state of hyperglutamatergic stimulation, you may have a neuron with a low dendritic arborization, a poor dendritic arborization, and much less synaptic buttons than the healthy situation. So if we are able to revert the trophic support and decrease in energy capacity that is induced by the state of major depression through interfering with the glutamatergic neurotransmission, we may be able to recover this aspect of the what we think is the cortex of these patients into this healthy picture. I have taken these drawings from a paper from Eric Nestler that were drawn in a different context, but I thought they were very useful. So this is the proposal, and because it's central, I will walk with you through all these points. So dysfunctional glutamatergic neurotransmission can result in activation and deactivation of intracellular cascades associated with trophic support and neuronal or glial cell death. I know that I'm asking you an act of faith now in believing that the increase of neurotransmission, glutamatergic neurotransmission, relates with atrophy, but I will show you some evidence about that. And decreasing glutamatergic neurotransmission in such systems may result in enhancement and maintenance of neural connectivity mechanisms which are essential for effective functioning, as I showed you in the slide before. But the intracellular downstream pathways may be targeted to produce a neuroprotective effect. In fact, it has been observed that the levels of glucocorticoid that are increased when you have an hyperactivation of the hypothalamo-pituitary adrenal axis. So when you are under a chronic state of stress, you have an activation of that axis, you have an increase of level of cortisol, and you have changes in the glucocorticoid receptors in the brain. So some of these receptors, there are two types, it doesn't matter. Some of those are associated with an increase in calcium currents and increase in NMDA receptor throughput, predisposing to neurotoxicity. So now we're having one of the hypothesized ways on which we can fall into depression bind to the fact that glutamatergic activity is increased in these patients who had an hypercortisol levels and actually changes in the activity of the glucocorticoid receptors. And then atrophy and possible death of some pyramidal neurons, the ones that are called in the area CA3 of the hippocampi, are at least in part the atrophy coming from increased glutamatergic neurotransmission. So this is the central hypothesis of what we're trying to resolve through a series of clinical studies. And here, what I have promised before is a paper that published a couple of months ago showing in a different context the importance of calcium regulation for the life of these neurons. So basically you have here a glutamatergic receptor with an increase in calcium levels in the intracellular, in the cytoplasma, but then this calcium in general is captured by the endoplasmic reticulum or the mitochondria, and then the neuron can survive these increases in calcium throughput. However, we are hypothesizing that there is a dysfunction in the capacity of store the intracellular calcium levels that are now elevated because of the hyperactivated glutamatergic neurotransmission. And actually, in fact, that has been associated with ruptures of the membrane of the mitochondria, and then this can cause There is an important protein called the permeability transition pore that gets disrupted when the calcium increases at levels that the mitochondria cannot manage, and there is a release of cytochrome C and the apoptosis-induced factor that may end up in an energy crisis and synaptic and neural apoptosis in the neuron. So I will not pay attention to this part of the slide, but here is central for the hypothesis. So, well, this sounds good in the paper. So it holds true or not in the clinical practice, which is probably what imports us the most. So I will share with you results of clinical studies targeting the glutamatergic neurotransmissions in patients with treatment-resistant mood disorder. And again, the same slide that I showed with you before, with the glia and the presynaptic neuron, the postsynaptic neuron, and the genotropic receptors, the metabotropic receptors, some transporters that I didn't mention before, and actually the pathways of synthesis of glutamate. But here I put the slide with new elements. So I have here two new drugs that I would like to pay attention. One is memantine and the other one is riluzole. I will start with riluzole. So riluzole is a molecule that is believed to decrease the release of glutamate into the synapse. Actually interrupts some voltage channels here, so decrease the release of glutamate neurotransmission. It is believed that may also have some function in postsynaptic receptors, but the main way that is believed to act is through the inhibition of release of glutamate in the synapse. So in the States, riluzole is approved for the treatment of amyotrophic lateral sclerosis, not for major depression or anything like that. It has neuroprotective value and evidence in animal models of Parkinson's disease, and stimulates nerve growth factor, including BDNF, brain-derived neurotrophic factor. We have mentioned before that some of the trophic support may be very important for the survival of neurons. And but here's the molecule. It has these are the usual parameters: protein binding of 96% and half-life of half a day. So we were able to show first in an open-label study. So this is a very small study, 19 patients. So please take this as a grain of salt in terms of the level of evidence. And it's an open-label study on which we treat patients with treatment-resistant major depression. So they have failed in average to more than 2 treatments before coming into this trial, this study. We have that 68% of the patients completed the trial, and we have here the final result. This is an open-label study, and you have that all the patients had a reduction in MADRS, was the main endpoint in this study, from 30-something, 33, 34, to 24 in total. So like 10 points, 12 points of decrease in the MADRS. And when you observe only the trial completers, those who were able to stay all the way, they had a reduce of around 18 points in the MADRS. Very impressive, but again, it's an open-label study. Let me show you with a little bit more of detail the data. So here we have the decrease in MADRS through the 6 weeks. And then all the patients are at the red line with a moderate decrease. For the trial completers, the decrease is even more important and noticeable, but very, very remarkable. For the responders, who by definition are the patients who are responding well to your medication, had an incredible decrease in this score. So from 35, 34 to 7 points. So although it's an open-label study, the data is very compelling that is something that this medication is doing in these patients. And I can tell you as an anecdote that the first patient was a patient that has resisted ECT and was the first patient, and probably the placebo effect was on us. We really wanted him to respond, and he responded incredibly. Nothing has worked before. It was an 18-year-old patient. So again, this gives you some level of evidence that there is some activity here between the glutamatergic system through the activity of riluzole in treatment-resistant mood disorder. So then we went to the next step, which was to investigate the same effect in patients with bipolar disorder who were depressed. This time, because bipolar disorder needs to be treated with a mood stabilizer, we secured that all the patients were receiving lithium for at least 4 weeks before starting the medication riluzole. So the plasmatic levels of lithium were therapeutic, between 0.6 and 1.2 milligrams per liter. And Again, a population, a trial that lasts 8 weeks, with doses of riluzole between 50 milligrams to 200 milligrams a day, the same that was utilized in the study that I showed you before with major depressive disorder. And these are the results. In patients with bipolar disorder who were already receiving lithium, we have a decrease between 33 and 18, something like 17 points in MADRS, again, very similar to the one that we observed in the unipolar patients. But for the trial completers, again, a robust decrease. And here, an important piece of information. It was mentioned today in the morning that approximately 30%, and I show you that in the second slide, of the patients are responding after they are being resistant to a treatment. If you increase the level of aggressivity in your therapeutic armamentarium, you can recover from the non-response between 30 and 40% of the patients. Here, these patients that were resistant to the treatment, who were on lithium, we recover them in a 50% of the cases when you take the response rate, which is, in general, for clinical trials, a decrease in 50% of the score that they came in with. But also importantly, the patients who reached remission were also 50%. So basically, the patients who responded remitted, which is an amazing finding. In general, remission is very low in clinical trials, around 20%. Caveats. This is an open-label study. Patients knew that we were receiving the medication, they know about the side effects, and the sample size is very small. But again, some evidence that riluzole, the medication that inhibits the release of glutamate in a synapse, may have a role in the treatment of bipolar depression and major depression when those are treatment-resistant patients. And here a little bit of evidence showing that the treatment also affects the anxiety symptoms. And also, I can tell you that in general, when you have a decrease in the depressive symptomatology, you have a decrease also in the anxiety. The pathophysiology may be shared. But the endpoint of these studies, the one that I showed you before, was the MADRS, the Montgomery-Åsberg Depression Rating Scale, that does not consider or includes, very importantly, anxiety symptoms, as the Hamilton Depression Rating Scale includes. And therefore, this is a different scale than the one utilized with the primary endpoint, and we have that Hamilton anxiety have, I think, a very robust decrease of around 6 points. In general, anxiolytic medications have a decrease of 4 to 5 points. So, again, open-label data. This is a post-hoc analysis. We're pooling population of patients, so a lot of care in making interpretation of these results. So that was the first set of studies, those two open-label studies. So then we move to a different medication, memantine, that different than riluzole does not act in the presynaptic neuron, but in the postsynaptic neuron, and here acts as an NMDA antagonist. NMDA, you can remember, is one of the genotropic channels that open for the entrance of calcium into the cell. So memantine is a poor antagonist, or weak antagonist, I would say, of the NMDA receptor. Memantine in the state is approved for the treatment of dementia, and we tried this medication this time in a double-blind placebo-controlled study. So, does the inhibition or antagonism on NMDA receptors have an antidepressant effect or not? So, here our answer. Now our sample size is increasing. We're having a better recruitment rate. 30 patients is very difficult for all of you who treat patients, it's very difficult to recruit patients for clinical studies. 30 patients with major depression received memantine between 5 and 20 milligrams for 8 weeks. They were washed out of prior medications, so they were treatment-resistant patients. We used the MADRS as the primary endpoint, and we can show you here the results. So in blue, placebo, and in red, memantine, and this is again MADRS, starting at 32, 31 points, going to 24, 25 in a double-blind study. So very small decrease. The delta here is not the usual that you can observe in antidepressant trials. But more importantly, placebo behaves better than memantine at endpoint. So the decrease in the MADRS score for placebo was better than the decrease in MADRS score for memantine. So this is a negative study. We don't have a sensitivity arm in this study, so we don't know if this is a failed study or not. but it's telling you that the effect is not as robust as the one that we observe open label, difficult to compare with Riluzole. But here we have that the NMDA weak antagonism does not result in an antidepressant effect. So we wanted to understand this a little bit better. So a study was designed with the utilization of ketamine. Ketamine is, as I told you before, is a potent NMDA receptor antagonist right in the middle of the channel. There are several antagonists that are used to compare potencies, and this is one of the most potent ones. And that is used as an anesthetic in humans, particularly in the pediatric population. But more importantly, when you give ketamine to patients, there is well-established increase in the euphoria state. I would say that actually increase in psychosis induced by ketamine. It has been used in a model in animals for the study of psychosis in humans. And here's the study. So this is a little bit, the design is a little bit different. This is a crossover study. So the same patients receive placebo or ketamine, or if they receive first ketamine, then they receive placebo. Only one infusion. Ketamine is available for intravenous use. It's not an oral medication. So we administer ketamine in the intensive care unit at the NIH and actually in the post-operatory unit. And the infusion was of 0.5 milligrams per kilogram, and it was done, if I remember correctly, between 30 and 40 minutes. So after the patients were free of treatment, all of them that were treatment resistant, they received an infusion in day 1 of ketamine or placebo, and then at day 8, they changed to the opposite, a crossover design. Here is the disposition of patients. Important to note that there is a wrong number there, but important to note is that patients who receive placebo first, in general, all of them receive ketamine, but the patients who receive ketamine first, half of them receive placebo, because the other half that received ketamine at the beginning did not got depressed enough to receive the placebo arm. So they had an antidepressant effect that lasts for more than 7 days, and I will show you that now. So the ages were the usual inclusion criteria, 18 to 80 years old, male and females. This included an adult population at this range. Good diagnosis of major depression without psychotic features. As I told you before, ketamine has been described to induce psychosis. And they have a moderate to severe depression. They were treatment refractory, and they didn't have substance abuse or dependence for 3 months. And this is the result. So when this result was published, I was not already in the lab, so I will share the data. It's very interesting data. So you can see here the scale, pay attention, is the decrease on the 21 items of the Hamilton Depression Rating Scale. And here we have the response in minutes. So before the infusion, 60 minutes before. After the infusion, an hour and a half, 2 hours, 230 minutes, day 1, day 2, 3, and 7. So basically, be careful here that the graph has logarithmic scale in time. But very, very interestingly, you have a huge decrease in the scores very early on. By 4 hours, you have a decrease of almost 11 points on the depression scale. When you compare with placebo, that had a decrease of only 5 points. And in fact, there is a statistical separation already there at the first 2 hours between the patients who were receiving ketamine and the patients who were receiving placebo. And that difference stayed throughout the study until the end of that period of 7 days. After that, they started to receive the other arm, placebo or ketamine. You may remember that half of them were not depressed enough to continue the study. They had to drop at this point when they received ketamine first. So very, very interesting. And of course, I have a lot of caveats on how you can blind this medication, and we can discuss that if we have some time at the end. And to show you here again some rate of response, again, very impressive proportion of responders, which is 50% of in this particular case of Hamilton Depression Rating Scale. And you have that ketamine compared to placebo, ketamine has improvements, response rates of near 70%, which is 50% was the study in Riluzole, the open label. 70% is robust response. You have to consider the patients, when they are receiving ketamine, they feel a little bit high, and therefore the placebo response is not easily separable from the antidepressant response. I will show you one slide of that thought. And again, the proportion of remitters, here you have the usual rate is in general around 25%, and here is 30%. As I told you before, for the real study it was 50% in the open label trial. So and here, getting close to the end of this presentation, we have that patients who are receiving ketamine have a high. They have an increase in the psychosis measured by the brief psychotic rating scale. But that high lasts only an hour and a half. At the 2 hours, you don't have anymore the high observed by the patients. And when you have the YMRS, which is a scale to measure manic symptoms, Young Mania Rating Scale, you have again an increase in the euphoria at the 40 minutes. By 2 hours, there is no difference between euphoria and no difference between psychosis effect of ketamine and placebo. And let me show you the back here. But 2 hours, you already have statistical separation for the antidepressant effect. Again, difficult to blind. And a lot of other caveats about this intervention. It's absolutely experimental, and there's not a recommendation to do it. It's just to share the data that potent blockade of the NMDA receptor may have an antidepressant response that may be dissociated from the psychosis induced by the NMDA blockage. So it will be ideal to come out with new medications that target this complex receptor system that may not induce psychosis. And there is a lot of ways to do this, and I will not stop here, but a lot of different intracellular pathways that can be targeted that interfere, particularly in this side of the slide again, the glutamatergic ionotropic receptors, and things that you can do in order to obtain this antidepressant response. I will not stop in this slide though, but to show you that opportunities are open and are there for the future. So just to share with you the summary of today's presentation. So we have reviewed that the glutamatergic system may have a role in the pathophysiology of mood disorders. We have summarized the preclinical evidence for the action of known antidepressants and mood stabilizers in the receptor, in the glutamatergic receptors. We have also discussed clinical supportive data demonstrating alterations of glutamatergic levels in patients with mood disorders. And we have reviewed the results of a series of clinical studies that targeted glutamatergic neurotransmission for the treatment of mood disorders, suggesting that this is a viable target for the development of new antidepressant interventions. And of course, that all of this work couldn't have been done without the patients and their families, their support. The originators of these ideas, my mentors at the NIH, Hussein Imanji and Carlos Arati, and my colleagues with whom we conducted this study, Jennifer Payne, Jonathan Sporn, and Jaskara Singh. The unit, the Mood and Anxiety Disorders Program at the National Institute of Health, and some funding that I received from NARSAT. And thank you for your attention.